Okay, this is the final recording for lecture 8. And I am going to just post this here. This is a challenge question. So you've got chromosomes here. Here's a pair of homologs. And that was not the right thing to do. Here is a pair of homologs. And here's another pair of homologs. And just think about the haploid cells that could result at the end of meiosis 1 without any crossover. You don't have to consider crossing over. You should come up with 4 different combinations, and so please do that as preparation for the lecture. And then meiosis 2. So in meiosis 2, this is another diagram that just follows up on the one I showed before that came from that Harvard website. And here we have, at the beginning of prophase 2, there are 2 daughter cells. Each one is haploid, but notice that there are the 2 different combinations. So these are the 2 cells that had resulted from the meiosis 1. These aren't the only combinations that could have been generated. Okay. During metaphase, the chromosomes, the spindle attaches to either side of the centromere that's on that one chromosome. This is exactly like happens in mitosis. And again, there's that equal pull towards the poles, and that lines the chromosomes up right in the middle. And as soon as they reach the middle, then some proteins cleave the connections between in this case, the chromatids, and those chromatids are going to fly apart the way they're shown here. And again, just if you see online or in textbooks actual images of chromosomes in the process of separating, this process of shortening and kind of reeling in the chromosomes towards the center through the microtubules is very rapid, and so the arms do really drag along behind like this. And then in telophase, new nuclear envelopes are formed around the chromosomes that are now chromosomes, not chromatids, because once they're separated from each other, they're referred to just as chromosomes. And then cytokinesis. So once the new nuclei have formed, then the cell's going to divide in half, and we will wind up now at the end of meiosis with 4 different daughter cells. Each one is a little different from the others, and they, due to the crossing over, for example, and allelic differences and so on. And these are not the only combinations possible. It depends on also the different combinations that could have existed up here. Okay, so this is just a rapid run through meiosis 2, because meiosis 2 is just like mitosis, except that the cell that's going through it is haploid, and we have had some crossing over, and so the homologs are not necessarily identical. They're not composed of 2 identical chromatids anymore. The chromatids of each homolog may be different from each other. Okay, and so this is just from one of the texts. I think this is from the Griffiths text, and this just shows it slightly oriented differently, but it shows basically the same things. How in meiosis 2, your cell is already haploid, but it's in the replicated state, and then at the end of the process you have 2 cells that are both haploid, and each of them has an unreplicated chromosome in there. And again, this is just a suggestion, but if you were to label one of the homologs, with a hashtag and another with a star or something, you could even just put nucleotide differences or allele differences, and you could see how the cells produced by meiosis are not genetically identical to each other. And you could go right back to meiosis 1 and start by labeling the homologs that way. Okay, so meiosis results in gametes that are haploid, or spores that are haploid in the case of fungi. And so you should hopefully understand at this point why this is necessary. And then it gives you genetically varied offsprings. The progeny they produce are not the same as each other, and they're not the same as either of the parents. And you should have some idea of why this is a desirable thing. And this is just a summary. So the things to know here are just basic terminology. The chromosome, the homolog, the chromatid, ploidy, replicated and unreplicated chromosomes should be added in here. And when we're talking about meiosis, which one is the reductional division and what does that mean? What is the equational division and what does that mean? And when I say that, I'm asking you to learn the terminology. What I mean is I am not looking for you to give me a memorized answer, but to explain in your own words what the term means, or if I show you a diagram, tell me what stage it is and how you know. Be able to label, what is a chromosome, what's a pair of homologs, what's a replicated, unreplicated chromosome, and or use those terms correctly when you're answering questions. That's the kind of evidence that we require to assess whether you do or do not understand the processes. And then the stages of meiosis and mitosis, there is more in the textbooks, in any textbook, than what I've mentioned here, but basically, I'm likely to show you a diagram and ask you which stage. I'm likely to show you a diagram and say, okay, if this is meiosis, what is the ploidy? If this is the starting cell with these particular chromosomes, what are possible outcomes of meiosis, or which of these diagrams is a reasonable outcome of meiosis 1 or meiosis 2, things like that. So it's not just memorizing and spewing out what happens at each stage, but knowing what that means by being able to draw it yourself, being able to interpret a diagram that I show you. If I show you a metaphase, tell me if it's meiosis or mitosis, and tell me how you know, things like that. The connection between an altered, sorry, that should have been underlined, not crossed out, between an altered DNA sequence and then how that gets passed on. So we're going to start talking in the next lecture about different ways of designating alleles and looking at how single genes segregate into the next generation and so on. So big A, little a, and what that what kind of phenotypes might be associated with that. And if you understand this lecture, you will understand kind of where those ratios come from better, and how the chromosomes replicate and then those DNA changes are passed on. Why meiosis is necessary if you're going to reproduce sexually, and why it's advantageous. What are the advantages of it, or the advantage? And then just be able to talk about meiosis and mitosis. What are they for? What are they used for? What is the purpose or the goal or the outcome of them? A little bit about the steps. Where are the steps similar? Where are they different? And those 3 key differences between meiosis and mitosis that I mentioned, and the stages that those occur at. Okay, and we'll see you in the lecture, which I can't even remember. I think it's going to be June the 3rd, I think, is when this lecture will be done. So I'm feeling better because I'm a little bit ahead of the class now. Okay, and we'll see you in class.